welcome to this panel, we have, as Alicia said, an absolutely stellar lineup, a full panel, so we'll try and keep it a very active discussion. The format that we've agreed is, this is very much going to be a storytelling panel. I think we all learn from stories, life lessons, innovations learned.
Now you tell me.
So that, oncology is probably, the area in biotech that's perhaps seen the most advances. It's the most partnered. It's perhaps had the most investment. All sorts of new modalities have come along that have really influenced the precision outcomes. So we're going to try and sort of touch on those points by individual panelists talking about part of the ecosystem that they have been in, the therapies that they've developed or not, the lessons learned along the way, and from that look at the new horizons and the sort of translational lessons to get a drug from that very early science ultimately to patients. So that's what we're going to try and achieve on the panel. Hopefully there'll be a bit of time for questions at the end. So first of all, we're just going to briefly introduce ourselves and how our experience and background is relevant to this panel. So my background is I'm more of a storyteller. So I've been in the biotech industry for over 30 years. So I've seen a lot of companies come, go, succeed along the way. Original background in science. I still love it very much. I'll pass on. Perhaps if we just go down the panel, introduce yourself.
Sure.
Along the line.
Morning, I'm Robin. I'm a medicinal chemist by background. I've spent about 10 years working in oncology in a couple of biotechs and a couple of decades in large pharma doing many different things. So delighted to be here today. Hope I can share something that's of some use or interest to you.
Hi everyone, so Daniel Marshall, so I'm Oncology BD at MSD, so I'm the Oncology lead in the London office covering Europe. In terms of my background, trained as an immunologist and then time in venture capital as well.
Hi everybody, my name is John Lyons. I'm part of the diversity in the panel because I'm Irish.
And I'm actually a drug hunter, I think.
By trade. I trained as a chemist originally and then moved into biological chemistry. And it's a nice story, but joined a number of biotechs in the US and the UK and have successfully helped launch a number of drugs.
Hi, I'm Neil Jones. I'm part of Cancer Research Horizons, which is the drug discovery and translational arm of Cancer Research UK. So I sort of represent that gap between the charity and the research sector and then moving into relationships and interactions with industry, which is where we focus a lot of our efforts.
Jolly good, thanks very much. I'm also an Irishman. I'm from Derry, but as you can tell because your ears aren't bleeding, I wasn't educated there and don't have the accent. I'm a pharmaceutical physician. I've been in industry for almost 30 years, did almost 20 years in big pharma, and then more recently I've been working with small companies, primarily between lead optimization and clinical proof of concept. My interest being in proving concept in the right clinical experiment in the right patient population. I'm very much a believer in Project Optimus, which was alluded to but the question with Project Optimus is how do we dose optimize, which I think is a very big question. I have academic roles. I have an honorary chair in translational medicine at Sheffield, where we have an interest not only in oncology, but also in maintaining health into old age, so multimorbidity and frailty, which are two other areas that are difficult to develop drugs in. And I also lecture to the students in Manchester, the poor students, that's what
Thank you.
Thank you. Yeah.
Okay, so as you can see, it's a panel that spans academia, industries, small biotech, big biotech, pharma. What we decided is as this is a sort of lessons learned and where are we going sort of panel, we're giving one panelist the opportunity to present one slide, and that's a more than 2-decade journey from spinning about a biotech company in the late 90s to reaching a point where 2 and nearly 3 drugs are in patients making a difference. So that's the whole journey from start to finish. So John Lyons from Astex is going to kick off with a 10 minutes at most, 8 to 7, case study of the Aztecs Journey, just to really set the scene of what the journey really is for translation to patients. Right.
Okay, so I will start. So the journey starts in 1999, you can see from the slide. The company was founded by Haran Jyoti, now Haran Jyoti OBE, Professor Sir Tom Blundell from the University of Cambridge, and the late Professor Chris Abel. So those three got together, they had a concept. The concept was quite simple. Well, at the time it was revolutionary, but it is nowadays quite simple. If I can crystallize a protein and I can do that robustly and reproducibly, then maybe I can use these crystals to find things that soak into the crystals. We nowadays call them fragments. And using this fragment-based drug discovery approach, I can slowly grow, and using chemistry, medicinal chemistry, I can grow the compounds in the crystal structure and see ultimately, after a lead-up program, some outcome, some positive outcome. In other words, I find new novel chemistry against a target that maybe people haven't been able to deliver or drug previously. So that was the concept. In 1999, Harren, as I said, founded the company with Tom and Chris in Cambridge. And Harren was a crystallographer. He did his postdoc in Oxford. And alongside Harren in Oxford was another guy called David Barford, who also was a crystallographer. David then went to the ICR after his postdoc. Harren went to GSK. And at ICR, David first of all worked on RAF. And once he had RAF done, he then worked on another a very good target in the RAS pathway called AKT or PKB. He called up Harren in 2002 and said, I've got the crystal structure, Harren. I'd really like to work together with you. So Paul Workman, who at the time was head of drug discovery alongside Michelle Garrett, who headed up the cell biology group, came to Astex and had a discussion about whether we could do a collaboration together. We, Astex, I mean. And that was the ICR-CRT collaboration that was initiated. I think it's in 2003. And I know that the year is right because in 2002, I met Michelle at a meeting in Frankfurt, and she was talking to a guy called Neil Thompson. After that, I applied for a job at Astex, and sure enough, Neil Thompson got my CV and said, yeah, I know this guy. I think we should talk to Michelle and maybe hire him. What happened at that time was I walked into the room, started talking about ureas, saying they were really good drugs, and a chemist, a medicinal chemist, sitting in the room here on the table, said to me, I don't think there'll ever be drugs. And that was Robin Carr. So Robin actually interviewed me and thankfully gave me a job. Yeah.
As I often am.
So before I came to, so we started a collaboration on AKT, which is very, very important to the company now. But before that, we also were approached by Novartis, and Novartis wanted to work with us on CDKs. We had already started up a pan-CDK program, but hadn't worked on CDK4/6. And when I was at Onyx, Pfizer and Onyx had worked on palbociclib, which was a CDK4/6 compound therapeutic, but it was without a crystal structure. So Astex were asked or charged by Novartis to work on the crystallography and did that successfully and moved that collaboration forward. So in 2000, I'm moving forward now to 2008, I think. In 2008, and I'll have to wind back again, on my second week at Astex, I was taken on a plane trip from Cambridge, from Stansted actually, up to Newcastle to meet Herbie Newell. And it was very, very cold. And on the EasyJet plane, everybody was wearing T-shirts, and I couldn't understand why they were. But now I know Newcastle, so yeah, I understand. So at that very first meeting, Herbie said, I think FGFR would be a great target. You guys should really work on it. So we chose not yet quite to work on it until it was validated. And Herbie did a lot of that validation work, and we trusted the work that he did. Okay. So that then allowed a collaboration to start between Newcastle and Astex, which eventually morphed into a collaboration together with Janssen based on the assets that Astex had derived from the collaboration with Newcastle. In 2009, based on the previous collaborations, I'm sorry, I just jumped over AstraZeneca, and I shouldn't do that, because in 2005, AstraZeneca talked to us. At the time, it was very difficult to get a crystal structure for AKT, so that was important that David Barford had done that prior to us signing a collaboration with him. And AstraZeneca came looking for additional assets for them to work on, and in the collaboration, that's what happened. We had some lead-up series, and they chose compounds and wanted to work on the program together with us, and then eventually only with us without ICR, and then by themselves afterwards. So in 2009, another collaboration with GSK, which is still ongoing. And all along, before I joined Astex, I worked at a company called Supergen. So Supergen was involved in in-licensing and forward development, and Supergen itself was a company that worked on hypermethylation or DNA methyltransferase inhibitors. And that company had this drug approved, made quite a lot of money, and then approached Astex about a merger because it allowed Astex at that time to become a public company. Once we became a public company, a company called Otsuka came to visit and said, we might be interested in doing something. At the very first meeting I sat there, and they said, we're not sure yet what, but we're quite interested in Astex. It's a nice company. And we said, that's not a collaboration that's going to go anywhere. And instead, actually, they acquired Astex for the drug discovery efforts, okay? So I'm looking here and I see that the work that Novartis did together with Bill Sellers was push Kisqali through as kind of a fast follower, but an improved compound compared to palbociclib. And they have now moved that compound not only into an approval position, but also into a very early adjuvant setting that is called NATALY, that just read out recently. So it's taken a long, long time for these things to deliver, but the NATALY trial is incredibly impressive from data that was presented recently from Novartis. And of course, I'll probably talk about MSD next, because we worked together with Taiho, and Taiho had assets in the RAS pathway, but they were not really accustomed to doing collaborations. So instead, we took the collaboration, we took the assets and discussed it with Catherine, Dan's colleague, and Catherine showed a lot of interest and said we should talk more. And so we signed a deal with MSD on the RAS assets, and then subsequent to that, added in an option for SHP2, which is an asset in that pathway as well. Meanwhile, AstraZeneca were working, as I said, on identifying a candidate separately post the research collaboration, and did development together with Ibanergy and then with Nick Turner at ICR, and beautifully delivered cabevasertib and a lovely phase 3 trial and an NDA that's now been accepted by the FDA. So it's looking really swell, I think is the best way to put it.
Thank you.
That's where we are.
So, I think you can see from that story the theme of the panel of how lots and lots of players in an ecosystem over a very long amount of time have driven that sort of innovation in precision oncology with many pharma partners, academic partners, charity partners, etc. So, I mean, as the new partner, perhaps, who hasn't yet delivered a drug to patients, but hopefully the collaboration will eventually. I mean, how's your experience of working with Astex and what you can expect in terms of that translation and collaboration along the way?
Sure. Yeah. I mean, I think this perfectly exemplifies how we like to work with biotech. Bringing that novel platform to the table and that novel know-how and expertise. And I think something that always strikes me about the collaboration with Astex is how culturally aligned the scientists are and how there's a really good dynamic there. And I know that's certainly perceived very positive internally. And I think when we're out looking for partnerships, which are, let's face it, absolutely key to our success. And I think over 50% of our revenue is driven by assets which have been sourced externally. So it's very crucial to our model. That's something we really look for. That it's the expertise you can bring to the table, but it's also that cultural alignment that allows the collaboration to be successful. And I think that's certainly evident from the ongoing collaboration at the moment.
Thank you. So moving from the sort of innovative platform to the other end, perhaps, of the journey and bringing the whole together. Neil, I think you're going to talk to us a little bit about, working at the more charity patient end of things and how that has gone, and use a couple of examples from your own collaboration in oncology.
Sure. Yeah, I mean, I think the role of the charity sector is kind of unique in that we can act as the glue to bring together a lot of different stakeholders, because ultimately the key stakeholders for the charity sector is the general public, so that's the patients, the fundraisers, the volunteers, so we can have a slightly different perspective, a slightly different view of how things need to go. A couple of areas I won't touch on a lot, but where the charity sector is really critical. One is around policy, and that's how we influence government and healthcare providers around the selection of medicines that can be taken forward, and also thinking particularly about things like earlier screening and diagnosis, because we actually know if we can catch cancer early, we can treat it a lot better, and a lot of medicines actually work if we can do that. So as you heard from some of the panellists earlier, charities can also help with clear, concise, available information to patients to help them to make informed decisions. And I think that whole patient advocacy piece has really been a theme throughout the earlier panels. And it's really critical that we see these people as partners in the journey of drug discovery. And that's probably a mindset change for the community as well. We're perhaps all guilty of often seeing the people that are treated with these drugs as our sort of test subjects, not partners in the journey, who actually can really inform and help us to design better medicines. The charities sort of really drive innovation. It's all about, obviously, the funding. And even in the current economic climate, the generosity of the public is still overwhelming. So it's still actually a good place where there is money available. But I think we need to be more creative in the schemes we use, and perhaps also try to break some of the cycle that associates funding only with publications, but actually drive a more translational aspect into how some of our researchers can take things forward. And actually also think about the researchers themselves. A lot of them are more entrepreneurial minded nowadays, so how does the charity sector encourage that and actually deliver, with those researchers so that they can actually take forward their ideas all the way to the clinic rather than just sort of disappear into the black box of drug discovery. So it's all about partnership. And also, as you heard from the panellists earlier, I think it's also about bringing in different parts of the research community together. So we heard earlier in the paediatric session about bringing developmental biologists into this approach. Obviously, we've seen with the successes of the immuno-oncology area and the drugs in that space, bringing immunologists and people who work on the fundamentals of the immune system earlier into cancer is really critical, and finding mechanisms and ways to fund that is a critical part. Something Cancer Research UK has done in that, obviously people are possibly aware of, is the Cancer Grand Challenges scheme, which is a really globally driven initiative, bringing the whole global community together with awards of up to £20 million over 5 years to tackle some of cancer's biggest problems. So previous awards have looked at things like cachexia, the microbiome, new treatments for paediatric cancers, and actually looked to the global community to bring forward ideas and proposals and fund them at the scale that is needed to deliver these. The first round of those have also started to read out really exciting impacts already. There was one around how we visualise tumours in 3 dimensions, which is actually helping surgeons really think about how they can treat tumours in a different way, how different surgery can work, and also new approaches to treatment. Another one looked at differential synthetic lethality, which again is providing some exciting new treatment opportunities. Jumping briefly ahead to the other end of the spectrum, we heard a lot earlier about clinical trials, and the role of the charities in this space is also really critical. CRUK funds experimental cancer medicine centres and a centre for drug development that can really help work with both the industrial community, our clinician networks, and research communities to really think about innovative trial design, sponsorship, and how we can take those forward in trial centres that are connected brilliantly with the NHS and also, again, with the global community. I think a critical part of this is also data, which people have mentioned in some of the other panels, and maybe the charity's role here can be to help act as custodians of data, because perhaps we're seen as a bit more neutral, a bit more trusted than other organisations. But again, I think it's how we involve the patients in that data journey and make sure that they are fully part of the process, fully part of the consent, and how we think about it. A really good example of that is, again, a few years ago, Charlie Swanton and a couple of other clinicians came to CRUK with the idea for TracerX, which is a massive screening and sort of longitudinal study of lung cancer right the way through the patient journey. And really that has had huge impacts already, and it's an ongoing study, even thinking about post-mortem samples as well, sort of tragically, but also really importantly, that those patients were prepared to give their consent for that so that we could actually learn from those samples as well. And that's had huge sort of impact into both our understanding of tumour evolution, heterogeneity, but also thinking about treatment responses, combinations and things, and really making impactful changes now to clinical practice, but also leading to new ideas, new modalities for therapeutic, and even new spin-out opportunities, investigator-led. I guess the final space I'll just talk about briefly is the translational gap, which is where Cancer Research Horizons mainly focuses. And really, we're about trying to bridge that space between early, exciting research that comes out of the networks and getting industry and biotechs involved, because we know we need the investment and the critical mass to really drive forward drug discovery. And I think there are 2 components that we really work to do there. One is how we engage our networks early in the drug discovery process and throughout the process, so researchers are really engaged and involved collectively throughout. We often, when we're looking at a themed area of biology, convene workshops and bring together the community to discuss and really brainstorm where the exciting opportunities could lie. Also, as you've heard about from John earlier, partnership early with industry is really critical. You heard about the cancer research technology, as it was then now, Cancer Research Horizons partnership with Aztecs, which is really bringing their shared capabilities together. And we've also done this in other areas with the likes of BMS, ONO, Merck KGA, in challenging areas of oncology, mRNA translation, immuno-oncology, the Hippo pathway. The other thing we're doing more of within Cancer Research Horizons is, again, recognising the entrepreneurial need of the researchers and also the way that spin-outs can actually accelerate drug discovery. And we've had some great successes with Iontura, which has taken 2 agents to the clinic, and also with Artios, just based on the campus here. We actually discovered the original pol theta inhibitor and then helped to operationalise Artios in its early days with our drug discovery scientists. And then as they grew their labs, it more moved into their sort of local labs here. So again, that early partnership is really critical. And I guess the last area that I'll touch on is, again, creative funding again, but think about how we engage the researchers into that early space. So something that we've really done in the last couple of years is created a new funding scheme called the Therapeutic Catalyst, where researchers and our in-house drug discoverers actually act as the co-applicants on awards that can really drive early translational activities. This can be target ID, target validation, or really that de-risking piece. We often see really exciting data, but to get it exciting enough for investors or partnerships with pharma, it just needs a little bit more, either to de-risk that you can actually make drugs in this space, or you can develop the right assays and things. So that award is really about bringing the community together to drive that. So I think in summary, the charity's role is really critical at driving that sort of cycle of bench to bedside and back again. And I guess my take home really is when we set up Cancer Research Horizons, we came up with the sort of tagline of further, faster, together. And I think that's really critical. This is a global problem. We need to work together. We need to therefore go further and deeper into the biology, the mechanistic understanding, so that then we can drive the development of new therapies much faster. Thank you.
Thank you. That's a great summary of all sorts of important areas. And I think you touched on the different modalities and new modalities that are coming through, and we've not touched on that yet. So I think, Robin, we're going to move to you next to sort of talk about how small molecules can perhaps morph into other modalities, and you're going to talk us about your experience at Merricks.
Yeah, so I retired about 4.5 years ago. I'm done. I'm just going to retire. My wife said, thank goodness, I'm sick of doing, 70% of domestics. And I retired for a summer, but a friend of mine made the mistake and he sent me a paper on a target called transferase, and he said, this is looking fantastic. Nobody wants to touch it because it's an essential gene. So if you inhibit it, you kill everything. That's like the very simplistic dogma. But if you dig in a level, there are some very important drugs that are against essential genes, but it comes with a big health warning. So you've got to have some real insight, you've got to have some reason you're going to beat the odds. And Roberto and Ed Tate and Andy Bell convinced me they did. It's a tractable target, so as a chemist that's very appealing. I was clear we could get a compound that was developable, within a year, 18 months. And the piece of data we generated, and it comes back to, I think, the ecosystem, was access to the screening at the Sanger. That an academic can get hold of 700 cancer cell lines and can get them screened very cheaply. And we put these little compounds in that Ed had, and they did not kill everything the same way. There was a spread of like 10,000-fold. Some cancer cells died very quickly at low concentrations, a lot of them in the middle, and quite a lot just didn't care. And so we thought there's more going on here. An essential gene is not an essential gene. There's subsets, you really need to dig in and understand it. And that hooked me. So I put my spade down and I jumped in, and they asked me, did I want to be a consultant or did I want to be part of the team? And the only way to do drug discovery is as part of the team. I mean, if you get to the stage of your career where you're happy giving advice and it's well regarded, fantastic. But the excitement of being with like-minded people to try and do something useful and not just earn a salary is unbelievably exciting. So there I was, completely convinced we would get a brilliant compound into the clinic. We can dissolve cancers with NMT. We can dissolve solid cancers. We can eradicate heme cancers. They're very well tolerated in mice. What is there not to like? And then you test them in rats, and they're not quite as safe. And then you test them in dogs, and you think, that's really tight. But here's this amazing efficacy. It's well worth the risk. And then you go out to investors and you say, look, we've got an essential gene, we've got brilliant compounds, we can dissolve cancers, blah, blah, blah. And they say, great, can you come back when you spent £10 million and you tell us whether humans are like the dog, which is bad news, or like the mouse? And of course, we weren't quite clear how to do that. And then we're a small team, but we network a lot. And one of the things we thought about was, why don't we take everything we've learned and take our small molecules, but rather than try and push it ahead as a single discrete molecule, we'll stick it on an antibody. We'll make an antibody-drug conjugate. Clearly, there's some spectacular successes. You hear it from patients, clinicians. It can transform the effectiveness of a tricky mechanism. And so we did that. We didn't know what the hell we were doing. You know, we had never designed, we'd never tested an ADC, and there we are thinking, we're going to pivot. We're going to become an ADC company. What do? You look around to friends and colleagues and you say, have any of you ever worked on ADC programs? And most of them said, what's an ADC? But a few of them said, yeah, I do. And a couple said, actually, I've invested in an ADC company and these people were great. Or I've got a friend that's worked on them. These people are great at testing them. And so we put together a virtual team. The generosity of those people working in this small niche area was just off the scale. You know, we just phoned up and said, we haven't got much expertise. You know, we haven't got much time. We really need to knock the ball out of the court. And they said, right, let's put an hour in the diary. And these CROs, contract research organizations, they put time and money, you know, their time into helping us think this idea through. We said, which antibody, which linkers, you know, we trust you. And we made them. And the results were spectacular. And we've gone on. So actually, it took probably the longest to convince our board to pivot, because this was a board that really believed in our first vision. They believed we were going to get small molecule drugs. They put money in. They went to their colleagues and said, it's worth investing. These guys are going to get small molecules. So actually, pivoting, thinking about how you pivot, is something I've really learned over the last 18 months, how much data you need to get, because you're asking people to let go of something they believed in. And that's often harder than getting new people to jump onto something because they're not invested in the past. And that's where we are with the story. So we're comparing our compounds now against the clinical leading ADCs. We've got clear superior differentiation from 3 marketed ADCs. We've got 10 out of 10 for efficacy. And now we're facing that, yeah, that's great, but what about safety? We've done an early study which looks very encouraging, and we're running a repeat 3 repeat doses cyno-tox study. Starts on the 17th of July. We will get the slides no later than the 30th of October, not that I'm time focused. And we presented to the AACR in the US, and one of the things I say that Astex did spectacularly well was realize you've got to be seen as experts. You know, you can be experts in this. You don't have to be experts in lots of things, but let the world know what you've really thought about. Let the world know what you need others to help with, but tell the world. So we put a poster at the AACR in Orlando, and I saw John there. At first time we told anybody we were working on an ADCs, and it was so uplifting to get feedback from 15, 20 different organizations. We're speaking to some of those to see whether we could or couldn't do a deal. That credibility, you know, you're probably at different stages of your career, but think about how you get known, how your team gets known, how you throw yourself out there to get really tough challenge from peers. And we've got tremendous feedback, and it's really changed what we're going to do over the next 6 months. If this story has a happy ending, and I like stories, and I like happy endings, we will go out to do a substantial Series A very early next year. Markets are tough, and then we'd look to do 1 or 2 corporate deals. We'll see. But it's a good reason to come out of retirement.
Thank you. I mean, that's a great story, and you can see the energy behind Robin's journey in taking this forward in an area of, you know, high science, high innovation, and Dan, perhaps you might want to jump in and comment. I mean, you're in ADCs in a big way, so I'm sure you guys will talk at some point in time.
Absolutely.
But,
In fact, I've got an email from one of your colleagues saying, can we have an updated slide set? And I'm just thinking, what's the sweet spot? You know?
Well, Dan might be able to give you a tip there, but, you know, I mean,
Clinical efficacy, no, can't do that.
how do you see the ADC space? I mean, it's, you know, one of the new modalities that's coming through.
Yeah, absolutely. I mean, it's really hot, right? I think you go to ACR, ASCO, whatever. There's a lot of posters, presentations on ADCs. I think you were probably pioneering the way pivoting to ADCs, because a lot of companies are doing that now, given the space and the funding environment for those sorts of companies. So yeah, I mean, it's exciting, right? I mean, I think it's purely been led by the really highly encouraging efficacy data in difficult to treat tumours. You only need to look at the responses in some of those agents, and you know, it's having a dramatic effect in some tumours. And obviously, it's a space we're very invested in. Yeah, I think there needs to be innovation to broaden outside the targets that everyone's focused on at the moment, and the payloads and the linkers, but that will come with time. There's lots of exciting stuff coming through from the grassroots up. So.
Oh, new payload here. So we're going to move on now, and I think it picks up on part of your journey that, that sort of toxicity. Is it like rats? Is it like dogs? Where do we go from here? And I know that's a point that you want to pick up on, Glen, in terms of that sort of preclinical area and the sort of dose escalation.
So the things that I've been pleased to hear are variations on collaboration, and it seems that people are talking at a much earlier time points than we would have done before. And then obviously the whole idea of safety and tolerability, and they are, as far as the patient is concerned, two potentially different things. We're going to get worried about a grade 3 neutropenia. The patient could be completely oblivious to that. But constant grade 1 nausea or constant grade 1 diarrhea is going to impact upon the patient. And we've heard that from the patients' groups for the last 20 years. But it's only really recently that I've heard that message back from the FDA, which is, yeah, your high dose might be the highest one that's tolerable, but 90% of the people that are taking your drug have got grade 1 diarrhea. You need to look at a lower dose. And out of that, as you alluded to before, is the Project Optimus guidance, which I shared the same, which is why weren't we doing this before? But we weren't. You go back to the aromatase inhibitors where we had an endpoint which is measurable, which was circulating estradiol, and Mitch Dowsett was clever enough for us to be able to measure that in a super sensitive assay. And we collected it, but we still chose too high doses to take into phase 3 and then dropped the one that was highest because it wasn't well tolerated. And that might be what we have to do with some drugs. But if you've got a targeted agent and that works in a particular patient population, then you should be thinking about how you can assess that target, both preclinically and clinically, both from a positive but also a negative effect, because inhibition of that target in normal tissues or over inhibition of that target in tumour tissues may impact upon the safety and the tolerability. So your translational package should be working up that pharmacodynamic strategy along with your patient selection strategy, because I'm all for diversity, although we still use Edwardian terms to define it. But to be slightly facetious, if my acid test clinical population is a white middle class heterosexual Catholic population, then that should probably be one of the early populations I should be looking at, because if I can't generate proof of concept data to get the investors excited and also the clinicians excited and the patients excited, then I'm not going to be able to get the funding to move forward. So what I need to do is to dial down that noise to signal ratio so I can truly understand the risk-benefit profile in the patient. And that probably requires you to have a very defined patient population. But again, as somebody else pointed out, if that is, middle-aged women with blue hair and a small dog, you're either going to have struggled to recruit your patients or you're going to have to go to a large number of different sites. So there's always a balance. There's always a balance with these things. But I think having this worked out preclinically as part of your target product profile, and most of the companies that I work with are writing their target product profile in parallel to their first-in-man clinical trial design, which is probably not the best way to do it. And then taking that out and talking to people. And yeah, we're good at talking to our investors. We're good at talking to big pharma if we want to sell our company to big pharma. The ones who are more sensible are talking to, the likes of Ruth Plummer.
We know. Yeah.
I know her very well. We trained together. She scares the bejesus out of me.
Me too.
But she's very bright. And these are individuals who understand translational medicine, but also understand how to then transfer that across into the clinic. Because I can design a clinical trial that is the perfect experiment. Yeah. But if she then hands it over to her nurse, and her nurse says, well, the patient can't be in ECG and MRI at the same time, you need that input. But then also you need the patient's input, and you look at the document that you're referring to, released at the beginning of this year. We've been waiting for it for a long time. Patient-reported outcomes are a key part of that. And when have we tended to do them? Probably half an hour before we submit our NDA document. But what they're saying is we want an understanding of the patient's journey through your treatment and the impact that has on their quality of life. And my understanding from the FDA is that they're actually open to the idea of using things that are not as well validated as we would have traditionally expected, the 40-page smiley face documents that we ask the patient to fill in about the previous month, or if you're a as one of my projects is, and you're in individuals with autism, you're asking the patient's mother to tell me how the patient felt over the last 30 months. And there are various tools to do it. Some of them are on telephones, some of them are wearables and what have you. And the FDA are open to this. The other regulators are as well. And we should probably be including them in our first in-man studies at an early time point. And I said to a statistician, but this is just going to add noise. And her response was, well, at least you've got the data. And machines are very clever these days, and I'm sure that they can do things with it. So working that translational package up as you are delivering your GLP tox data, delivering your DMPK data, delivering your fine-tuning and writing that into some sort of document, a target product profile, getting the tires kicked on that very widely, and then you can move into designing your clinical trial. And my preference is to develop a clinical trial remit document, design remit document that I then circulate. And then when everybody has fed back on that, I lock that, and then I hand that to the medical writer to produce the protocol. Because otherwise, if people are reviewing a protocol, the medical writer is slowly going insane as things are changing. So that's how I see translational medicine, and I think that's probably valid across a number of different treatment modalities. And maybe the last thing I can do is to talk about one project I'm working with Helen McCarthy from Queen's on, where she has a novel way of delivering mRNA vaccines. And we were talking to Esther Moss in Leicester about potentially moving this at a very early time point into women with cervical intraepithelial neoplasia. And she said, do you realise that the majority of women who have HPV-driven disease are probably from a low socioeconomic class and a recent arrival within the UK? And if there's two things they don't like are vaccines and clinical trials. So we're actually working with her to understand how best to approach those patients and include them in the clinical trial, because, you know, if I was an investor, I might say, well, go to a paternalistic country and recruit your patients. And you could do that, but at some point we're going to want to give this vaccine to patients within the UK, so we need to understand our market. And that is being funded by the investors of this company, because they get the fact that we need to do that, as opposed to the response that I've often had, which is, don't those data sit on the shelf? Can't you get them out of a publication? And I think that's the sort of thing that you need to think holistically with regards to your translational package, because it is what's going to drive the design of your early clinical trials. I'll be quiet now.
Thank you. So we had one more point that we were going to sort of talk about as a panel before we open up for questions, and that was really the whole concept that I think, particularly with new modalities and sort of new areas like precision oncology, of who holds the baton, who's the hero along the journey to get it from the start to the finish. And I think we've heard stories of you know, the patients are key in this, the collaborators are key in this, but pharma's key in this as well, because ultimately it's going to be pharma that takes the drugs to market. So perhaps, Dan, if I start with you talking about that, you know, at what point from the academics, the charities, the biotech companies, in these sort of new areas, particularly in the new modality areas, you know, how do you pick up that baton, and how does the sort of risk appetite, I think, align? You know, I think biotechs and you know, people like Robin have a very high risk appetite because they're very excited by the new things, but by the time you know, the baton gets handed on, you know, how does that relationship work? How does the process work? And
Sure,
perhaps a few stories from your side would be helpful.
Yeah, absolutely. I mean, in terms of the risk appetite, I think, you know, early on, I would say the risk appetite is far higher. Obviously, you know, as you progress through the process, things get gradually more expensive, right? So inevitably, when you're deploying that level of capital, you have to have a different assessment of your risk. Okay, I would say, you know, at MSD, the precision oncology side of things is very central to what we kind of look for. So we really like to know what patient group we're going to go in, and that informs, you know, all the way through discovery, through clinical development. To know exactly which patient group you're going to position your agent in is key, and it's a key fundamental part of even the first pass diligence. So obviously, you know, inherent in the precision oncology approach is you're considering that up front, right? I think you have to look at, well, I mean, there's numerous examples out there of, you know, various point mutations or, you know, insertions within key oncogenic drivers. So if you know that up front and you can design your strategy around that, And that's fantastic because you know where you're going to position it and you can see it all the way through essentially that development strategy. It's also relatively de-risking as well. I mean, you know, with the advent of new technologies, it's far easier to interpret defects within the cell machinery that are often these oncogenic drivers. So it can be de-risking from a kind of target basis as well, which is nice. Obviously, you know, it's not a slam dunk. It's far more complicated than just knowing, you know, where your mutation is. I think there's numerous examples out there of, you know, agents which don't work in the mutant population where they should. So, I don't know, BRAF inhibitors, right? So they don't always work in BRAF mutant tumours, and it depends on where the tumour is. For example, I think in that particular case, it's EGFR feedback, exactly, right. So, for example, in colorectal cancer, where EGFR signaling is relevant, you know, you don't get the responses you should. So, I think you can consider the precision oncology approach and it being de-risking, but you have to consider the context of the biology as well. So, I mean, that's a key consideration, you know, when we're assessing risk to bring in new assets and take them through the clinic. I mean, I think another key one in the precision oncology side of things, and precision medicine in general, actually, you know, that's not just necessarily around this down to oncology, it is actually the prevalence of genetic testing out in the clinic and populations as well, and testing in general. I think there's, you know, it has hampered the commercial rollout of some drugs, that some of this commercial, some of this testing hasn't been as widely available as perhaps anticipated when those agents were launched. And obviously there's countless attempts by different companies and charities to try and help address that. So we can roll these out. And that speaks to development process as well with regards to clinical development. So obviously, I mean, if you're going into pretty rare mutations, you know, sometimes if you're going into a very super rare subset of a particular tumour type for a particular point mutation, you know, you need the prevalence of that testing to recruit for that trial. And obviously that speaks to the commercial picture as well. My commercial colleagues are regularly shouting at me for looking at things where perhaps the market at the end is too niche. But you know, that doesn't stop us, right? We're very focused on unmet need, and you know, I think if we could, given a de-risked approach, launch those agents into those populations, that certainly doesn't stop us. I think another consideration is the actual development of them. So we've kind of touched on the development, but you know, I think if you start to look at maybe, say, for example, tumour agnostic approaches, where you're developing agents within, you know, tumours from various different tissues, then, you know, that can be kind of difficult sometimes for me. I mean, it's not my area of expertise, but it can be difficult when you're kind of assessing that data, right? It's often a single arm, no control arm. You can't randomise, especially if you've got a high response rate, you can't randomise patients off that, right? So it's single arm and can be hard to assess. So that's kind of another consideration, and it speaks to that balance of that kind of risk-benefit. But I mean, looking at everything holistically, it's a space that we're very interested in. I think a lot of pharma are very interested in, particularly the kind of synthetic lethality side of things as well, where you can exploit genetic vulnerabilities for your gain. Yeah, and I think it's de-risking overall.
Thank you. So I think, Alicia, we've got 5 minutes or so. So if the panel's happy, shall we have a discussion with the audience if there's any questions? I think there's one here at the front.
Am I the first one?
Yep.
Okay. Now, I was particularly interested by the career path of Robin, because like me, I was a medicinal chemist. I worked on fragments, and I passed to the ADC afterwards. And I was interested, well, later I will ask you questions about the technology platform that you use for your link. But I wanted to do like a more generic questions. So I just wanted to ask you, how do you see like the future of small molecules compared to biologics? We will have like more and more biologics, therapeutics in the market, and small molecules will just slim down in the future. What do you think?
I'm married to somebody that spent years at CAT and MediMune, and she felt my passion was too deep and myopic for small molecules. And then I ended up being accountable for small molecule platforms and antibody platforms at GSK. And it was a real eye-opener. How for antibodies, you get certain things just, like maybe 70% of your process just repeats. Your PK is pretty standard, your QC readout pretty standard. So it's like an engineering plus some bits of novelty. So I guess my learning is be more open-minded than you think you've got time to be. Keep checking your prejudices and try and buddy up and listen to people that have used those other areas. Because actually, as I'm coming to the end of my career, those sort of non-obvious bits have been the bits that have been most fun, and actually people that I've got to know that have actually they've bothered to spend time with me. So actually, I'm absolutely convinced there's a long-term future for small molecules in terms of helping healthcare. Whether they're 90% generic, I don't care. Whether they're less profitable, I don't care. But small molecules are cheap to manufacture, they're simple to store, you can get them out throughout the world. And actually, if you look at the life expectancy increases we've seen in the last 70 years, you look at the drugs that delivered it, something like 90% of those are now generic. So it's like job done. So I'm not sure the need for innovation in small molecules is as compelling, but the value is really high. And I think it'll be those interfaces.
Yeah, I mean, if I could kind of touch on that as well, I think we're seeing new generations of small molecules come through, right? Look at PROTACs, molecular glues, cyclic peptides. Is that a small molecule? I don't know. Everything in between, right? We're seeing a blurring of the modalities. I think it's, I personally don't sign up to the fact that small molecules are going to disappear. That, I mean, just look at some of the PROTAC stuff, right? That's coming through. There'll be innovation. The IRA may have a deciding factor on the investment thesis behind one versus the other. Who knows? But yeah, I think there's a place for everything in the right context. Yeah.
I think the world's better at doing antibody research and development than small molecule. Small molecules, is so incredibly bespoke. There's so many issues. You cannot predict the off-target activity at all well. So many small molecules fail.
The risk profiles are just different, right?
Yeah, and I guess Astex is not static either. We've of course moved more to using cryo-EM. So we have our own microscopes, which allows us different insights. And just for finding out where are the goalposts when you're proposing a target. For example, you have academic collaborators who tell you what they think is good, and then you have big pharma partners who say, well, that's not good, I'm not going to do that. The mission, the choice we make is always, can you do a trial with less than 100 patients to understand if your drug is working or not?
Thank you. And I think, the small molecules are operating via different mechanisms of action. It's not, the old-fashioned sort of drug target. There's synthetic lethality via the microbiome, so it's actually growing in complexity.
So an example would be MET as a target. The monoclonals just didn't work, but the small molecules did. Another example might be where you only require a drug with a very short half-life, so some of the DNA damage repair targets. You just want to walk the patient past the bottle, in which case a monoclonal is not going to play to that.
Yeah.
Or advanced disease, where highly selective tyrosine kinase inhibitors that we developed at AstraZeneca didn't work in advanced disease because they were too selective, whereas drugs like dasatinib, which were pan-kinase inhibitors, had activity because of the heterogeneity of the disease in the patient. Yeah. But it's horses for courses is always the case, which I think is what everybody
Did we have one other question at the back?
Still good?
I think this is going to be our last question for the panel.
Thank you. Michael Tarn from Precision for Medicine, clinical CRO. So, thinking from what Neil had mentioned earlier on, maximum tolerated doses, but actually looking at other areas in terms of AEs, etc., and touching on Project Optimus. The 3+3 model, tried, tested, it's great. But are you, and any of the panellists really, seeing an uptake with the Bayesian models, Boyne design, etc.? And if not, is it because there's a lack of awareness, and whether that's from a biotech standpoint and or a principal investigator uptake?
I'd quite like to answer this question, because I always say and I will say the same to you, I want to be converted to Bayesian designs. And I had just prior to the cup match in Dublin, the rugby, I was surrounded by some Bayesian converts, and while we were waiting for the rugby, they spent an hour trying to convince me. I don't necessarily think that it fits in with Project Optimus, because at the moment we tend to use toxicity and safety as triggers to expand the cohorts. Okay, now what you could do, which would make a lot of sense, is to say, well, what we'll try and do is use a clever machine to use a mixture of what we want the drug to do and what we don't want the drug to do to be in the model. But then I'm getting my head pecked by the CRA because the time until that dose escalation meeting has been pushed out to 2 to 3 months rather than the 3 weeks that they wanted. Okay, so I really do get it. And I think if you've got a drug with a narrow, very narrow therapeutic index preclinically, Bayesian design is probably going to be what you want it to do. A lot of my drugs are squeaky clean in animals, which doesn't mean to say they're going to be squeaky clean in humans. So what I tend to do is use what Stefan Simonides at Edinburgh describes the rolling 4 design, which is a modification of the rolling 6 design, where you recruit 4 patients to each cohort just in case one of them falls over. So you've still got 3 patients. And I will define a minimally biologically active dose based either on exposure, if we can't work up a pharmacodynamic biomarker, or ideally something as close to proof of mechanism as possible. And that's telling me where I've moved from homeopathy into the drug doing what I want it to do. Now, the FDA have told me that they're highly suspicious of MTD, and that with small molecules, many of those drugs that we define an MTD and what we used to call phase 1 turns out not to be the MTD. Gives capecitabine, nivolumab, various things as very good examples of that. But I quite like to know what happens if the patient takes too much of the drug, so I'm going to define an MTD anyway. But what the FDA have made it clear to me when I've discussed their document that you should read, if you haven't read it, because it's how we should do drug development, or at least the philosophy behind how we should do it. They don't tell us how to do it, they just say what we want. Is then I will build the ability to be able to backfill or expand some cohorts that sit somewhere between your minimally biologically active dose and your MTD. And I could build in a Bayesian aspect to that to allow ourselves to be able to open that. I don't understand it, so I hand that to a statistician. It's a bit like black magic. But then that allows me to do that, either based on something that Bayesian can work with, or just because it's interesting, just because we saw a response in a patient with a sarcoma. And then I can expand that. And then that allows me the ability to be able to deliver what I want and what the regulators want. The problem is, when I sit down with the investigators, they see that's too expensive.
So I think...
Sorry. Thank you.
Yeah. With that comment, I think we're going to have to close there. So I would just like to thank conference organisers for inviting us all onto the panel. Thank very much the panel for their insights, thoughts, contributions. And thank you very much for being a patient audience.
Thank you.